Hi everyone, this is Jonathan Cedillo, student at Grand Canyon University, and today I'm presenting to you the effect of teething strain in the Frank-Starling law involving thin and thick protein filaments. Presentation for the class, Biology 520, Concepts of Human Physiology 2, Dr. Joan Tassoni. The following paper presented, Teething strain contributes to the Frank-Starling law of the heart by structural rearrangements of both thin and thick filament proteins, emphasizes the Frank-Starling mechanisms of the heart. Such event is correspondent in the modulation of the myofilament calcium sensitivity by sarcomere length or length-dependent activation. Further research needs to be done to understand this mechanism, but studies have revealed that the large protein molecule titin has a role in the length-dependent activation. The theoretical role of titin is that it intervenes to approximate myosin heads to actin molecule. Experimentation was carried on to identify the exact role of titin in length-dependent activation by utilizing myocardium of rats. A comparison was observed between wild-type and homozygous mutant myocardium of rats. The observation showed that after muscle tissue is stretched, the homozygous mutant had less passive force than a wild-type muscle tissue. After analyzing the wild-type twitching muscles, the results demonstrated that after lengthening the muscle, myosin and troponin activity was upregulated as well as a decrease in cross-bridge space activity. In addition, other probes demonstrated that there was an increase in the mass and organization of thick and thin filaments. Also, sarcomere length modifications were found in wild-type myocardium but absent in the homozygous mutant. Finally, results of the experimentation showed a link between titin protein strain and the Frank-Starling mechanism. Everything points out that the mechanism in this event shows no evidence in the involvement of interfilament spacing or movement of myosin toward actin. However, the lengthening in the sarcomere changes structure in thin and thin filaments that have a correlation with titin strain molecule and myofilament of the length-dependent activation. The Frank-Starling law of the heart talks about the mechanisms that regulate a beat-to-beat basis of the heart. The authors of this article established that there is a link between the ventricular end systolic volume and end systolic pressure, which is dictated by contractions of the heart. This is suggesting that the ventricular stroke volume is directly proportional to the extent of diastolic filling. The heart rate and contraction activity of the same allows the Frank-Starling mechanism for most of the cardiac activity. However, more studies need to be done to fully understand the Frank-Starling process. Furthermore, the upregulation of the sarcomere length allows for twitch force development. Studies in isolated myocardium suggest significant results regarding the upregulation in calcium levels that influence the sarcomere enlargement in response to contractions of the heart, event which is known as a myofilament length dependent activation or myofilament LDA. Recent studies reveal that LDA is a relatively fast process followed by a modification in the sarcomere length, which can be dictated by strain-dependent mechanical rearrangement of contractile proteins. LDA is said to have a greater activity in the myocardium than skeletal muscle. Also, research proves that cardiac LDA is regulated by contractile protein phosphorylation together with disorders in the heart associated with mutations of several proteins involved in heart contraction activity. Additionally, authors of the paper state that the passive force is initiated by titin protein from the sarcomere that influences the myofilament calcium responsiveness. Further, rat animal models shows the natural process that represents a mutation in the splicing factor RBM20, making a modification in titin mRNA splicing. A possible outcome of this mutation is the synthesis of the large protein titin isoform in the homozygous mutant myocardium. The presence of the isoform molecule in the homozygous mutant heart muscle showed a downregulation in the cardiac passive force after stretching the muscle tissue, as well as blunted Frank-Starling response and reduced myofilament LDA. Time-resolved small-angle X-ray diffraction technique was used to confirm conformational structure changes in thin and thick filaments contractile proteins at relaxed phase of cardiac cycle in wild type only. Data show that tetin strain had a significant effect in the cardiac Frank starting mechanism. This information suggested that interfilament spacing or movement of myosin heads toward actin in the relaxed muscle is not involved. However, after stretching the muscle tissue, it appears that there are structural modifications in thin and thick filaments that are regulated by titin strain. Now, results of experimentation. With this figure presented, authors of the article explain the impact on muscle function of the twitch force from the wild type and homozygous type myocardium of rats. The tissues were stimulated electrically at relative short sarcomere length and long sarcomere length. Prior running the electricity over the myocardium, the tissue was quickly exposed to X-rays shown by the blue bar in figure 1A. This process was performed 30 times every 10 twitches. The two types of rat myocardium stretch induce an upregulation in the passive and active twitch force. However, there was a reduction in the force of the homozygous mutant myocardium. Figure 1b presents a normal twitch force upregulation after stretching the sarcomere. Stretch induced showed approximately 320% of starting twitch force in wild-type myocardium in comparison to the 134% force of the homozygous mutant muscle tissue. This data is suggesting that reduced titin strain is related to a compelling reduction of the force in the Franklin-Starling Law mechanism. In figure 2, authors of the paper present and explain the impact on 2D X-ray meridional reflections. These patterns were recorded during short and long sarcomere length in the wild-type heart muscle. The figure shows that the meridional reflections are horizontal. These come from thin and thick filament proteins such as myosin, which are labeled as M1 through M6, and troponin labeled as TN1 through TN3, and myosin binding protein labeled as letter C. This last protein shows to be in pairs, which suggests the interaction of two sarcomeres. In figure 2a, we can see that there is presence of an increase in stretch-induced meridional reflections. Additionally, figure 2b presents the meridional reflections of the wild-type and homozygous mutant at the top and bottom respectively. Also, it shows the myocardium at short and long sarcomere lengths respectively as well. Only at the wild-type myocardium model, there was evidence of change in the sarcomere structure after elongation. Further, the proteins myosin, troponin, and myosin binding protein showed a compelling upregulation of activity after the myocardium of the wild-type model was stretched. However, this was not the case for the homozygous mutant. In addition, after a stretch factor, the rhythm of myosin and troponin was increasing in the wild type but not in the homozygous mutant. Here, in figure 3, authors of the publication show the representation of the impact on 2D X-ray myosin layer lines. Data specifies the first-order myosin layer line projections from short and long sarcomere length of wild-type and homozygous mutant myocardium. Figure 3a is a 2D X-ray that shows the myosin layer enclosed by the rectangle. The location of the myosin heads can be identified from the position of the first intensity maxima along the layer line. In general, the location of cross bridges was approximately 19% ahead of the thick filament in wild type in contrast to the homozygous mutant myocardium. Further, sarcomere elongation of the wild type muscle tissue located myosin heads approximately 8% closer to thick filaments, but there was no movement activity of the homozygous mutant muscle tissue. Figure 4 shows the impact on equatorial reflections at short and long sarcomere lengths of wild type and homozygous mutant muscle tissue. Figure 4b at the top graphs, there is a representation of the lattice spacing. At the bottom of the figure 4b, we can see the intensity ratio between 1,1 and 1,0 plane of symmetry. Surprisingly, elongation event reduced the spacing between lattices of the wild type and homozygous mutant muscles. The approximate lattice spacing across the muscles was about 2%. However, homozygous mutants showed more compression at wild type muscles. As already mentioned, intensity ratios were reduced in both groups after stretch. In the homozygous mutant group, the intensity ratio was approximately 12% smaller than the wild type group. This intensity ratio is the movement of myosin towards actin molecule. Even though contraction activity of the myofilaments length dependent parameters of both groups, wild type and mutant homozygous, of the myocardium were different. There are similarities of lattice spacing and intensity ratio between the two groups. This is suggesting that none of the parameters of lattice spacing and intensity ratio are related to myofilament length dependent activation of LDA, as mentioned at the beginning. After acquiring information presented in the previous figures about the sarcomere structure, authors of the paper claimed to have reconstructed radial projection electron density maps. The electron density maps for short and long sarcomere lengths and the difference map between the short and long sarcomere length of the wild type and homozygous mutant myocardium are presented in figure 5. In the wild type group, there was a significant upregulation in electron density of both filaments, possibly due to the structural organization of the thick and thin filaments located at their respective lattice position, but there was no compelling changes in the homozygous mutant muscle tissue. In addition, after a stretch and enough abnormal electron density, approximately 25% of exceeded the peak thick filament backbone density, which was noticed in both filaments of the wild type muscle. This is pointed with the yellow arrow of figure 5 at the bottom right. In figure 6a, authors of this publication showed activity of myosin during attachment. Figure 6a also depicts the transmission channel at the top, the red phalloidin at the middle, and green TNC at the bottom. A larger scale of these same pictures are presented on the right side of figure 6a in conjunction with the colocalization of TNC with actin. In figure 6b, authors explain that the myocyte TNC fluorescence went back to normal sarcomere length due to calcium activity. Furthermore, only in wild-type myocardium stretch events upregulated TNC fluorescence. However, upregulation of calcium led to a decrease in TNC fluorescence. Interestingly, calcium sensitivity is not affected by sarcomere elongation. Another relationship of upregulated calcium sensitivity was noted at homozygous mutant myocardium, regardless of the inactivity of other variables after stretching events. Moreover, elongation in the wild-type myocardium tissue dictated a conformational rearrangement of troponin, but not in homozygous mutant myocytes. The main intention of this research was to apply a small angle X-ray diffraction of intact twitching myocardium from wild-type and titin mutant rats to analyze the activity in the sarcomere to the regulation of myofilament length-dependent activation. Authors hypothesize that by analyzing different locations across myosin heads during the stretching events, we'll conduct a higher number of actin-myosin activity at the diastole and twitching muscles that could represent evidence over the influence of myofilament length dependent activation. However, results of the experimentation point out that this was not the case. Myofilament length dependent activation is not affected by the radial outward movement of myosin heads towards a thin filament after stretching the myocardium, as was suggested before. Additionally, the results suggested that cross-bridge activity in conjunction with the thick filament backbone move after diastolic stretching in wild-type myocardium, but authors of the article could not say the same for homozygous mutant muscle. Furthermore, teething mechanical strain leads to the myofilament length-dependent activation, demonstrating that myofilament LDA has a significant activity in homozygous mutant myocardium, but only when the sarcomere length is relatively stretched along distances, on where passive forces are accountable in these types of myocytes. Further, scientists explain that the RBM20 mutant only affects the teething length at the cardiac sarcomere. The results of the experimentation demonstrate that there is a mechanism where teething strain affects the thick and thin filaments, which leads to actin myosin interaction, hence allowing for more strength at the longer sarcomere lengths. To conclude this presentation, authors of the publication state the following. It is clear from our current results that we can conclusively rule out altered interfilament lattice spacing as well as close approach of myosin heads toward actin at longer sarcomere lengths as being responsible for myofilament length dependent activation. Thank you for watching. See you next time.